Welcome to Service Scholars Academy. Now, we have highlighted the importance of studying and how you can study in biology and how it is important to understand the terms that you're going to be using and how to use them each and every time when you are writing an examination and how you should be revising. So now we are going to get into the cell structure, and we are first starting with microscopy. So we have the light microscope and we have the electron microscopes. So these are some of the microscopes that we use when we are dealing with cells. So first we are going to define what a cell is. So cells are the basic unit from which organisms are made. Most cells are very small and their structures cannot be seen with a visible eye, but can only be seen using a microscope. So at A level, you're most likely to use the light microscope. So light rays pass through the specimen on the slide and are focused by an objective lens and the eyepiece lens. This produces a magnified image of the specimen on the retina of your eye. Alternatively, the image can be projected onto the screen or recorded by a camera. An electron microscope uses beams of electrons rather than light rays. The specimen has to be very thin and must be placed in a vacuum to allow electrons to pass through it. The electrons are focused onto the screen or onto a photographic film where they form a magnified image of the specimen. So when you are using the light microscope, you know that we have light passing through. In electron microscope, we have electrons passing through. So now let's look into the magnification and resolution. So magnification can be defined as magnification is equal to size of image divided by the actual size of object. So this can be rearranged to actual size of object is equal to size of image divided by magnification. So there is no limit to the amount you can magnify an image. However, the amount of useful magnification depends on the resolution of the microscope. This is the ability of the microscope to distinguish two objects as separate from one another. The smaller the objects that can be distinguished, the higher the resolution. Resolution is determined by the wavelength of the rays that are being used to view the specimen. The wavelength of a beam of electron is much smaller than the wavelength of light. An electron microscope can therefore distinguish between much smaller objects than a light microscope. In other words, an electron microscope has a much higher resolution than a light microscope. We can therefore see more fine detail of a cell using an electron microscope than using a light microscope. So as cells are very small, we have to use units much smaller than millimeters to measure them. These units are micrometers and nanometers. So 1 millimeter is equivalent to 1 times 10 to the power negative 3 meters. 1 micrometer is equal to 1 times 10 to the power negative 6 meters. And 1 nanometer is equivalent to 1 times 10 to the power negative 9 meters. So to change nanometers or to change millimeters, to change millimeters to micrometers, multiply by 1000. So this is when you want to change a micrometer, you want to change a millimeter into micrometers, so you multiply by 1000. So magnification calculations. So you will be able to work out the real size of an object. So if you are told how much it has been magnified, you can be able to work out the real size of an object. For example, you can have a drawing of a mitochondria, right? That has been magnified to 100,000 times. You can use a ruler to measure the length in millimeters, and you can find, if let's say you find it to be 50 millimeters long. Now, as it is a very small object, convert this measurement to micrometers by multiplying by 1000. So you take the 50 millimeter, you multiply it by 1000 to get 50,000 micrometers. Then you substitute it into the equation. Remember, our equation was actual size of the object is equals to size of the image divided by magnification. So for the size of the image, we multiplied it 50 millimeters and we found out that it's 50,000 micrometers. Now it has been magnified 100,000 times. So on magnification, we then put 100,000, which gives us 0.5 micrometers. So you can use a scale bar to do a similar calculation for a drawing even of a what? Of a microscope, of a chloroplast. So you measure its length using a scale bar, right? Which you will be given. Maybe let's say the scale bar is around 2 micrometers. So you can be able to then calculate that. So to then come up with magnification, you calculate its magnification using the formula. Magnification is equal to size of image divided by actual size of the object. So you are looking into the length of the scale bar, and the length of the scale bar divided by length of the scale bar represents, right? So which is 200,000, right? 200,000 divided by 2,000 divided by 2. So when you then find, you then find 100,000. That is magnification of 100,000, where you divide 2 into 200,000, where you get 100,000. That means the chloroplast has been magnified 100,000 times.